Welcome to Chapter 4. This is going to cover just a small snapshot of eukaryotic cells because our primary focus for this course is going to be prokaryotes. But we'd be remiss not to cover at least a little bit over eukaryotes, just not the entirety of Chapter 4. So let's go ahead and tuck in and get started. It's important to understand the primary features of eukaryotic cells. You can probably imagine from various classes that we're talking about animal cells, possibly plant cells. Those are our primary focuses of eukaryotes. The big important features to remember are the following. Eukaryotes are going to be much larger than prokaryotic cells, and that's because they're going to contain all of those different organelles, which prokaryotic cells lack. They do not contain peptidoglycan, and their form of reproduction is going to vary depending on what type of cell or organism we're talking about. So the two flavors of reproduction include asexual or sexual, which we'll talk a little bit about here in a bit, but those are our primary features we need you to know about eukaryotic cells. There are two types of appendages. We've already talked about these when it comes to prokaryotes, but let's go over them again when it comes to eukaryotic cells. We've got the flagellum, which are going to be much fewer and longer and thicker on eukaryotic cells than on prokaryotes, and they're going to move in a wave-like motion, while cilia can be many and short are going to move in a back and forth motion. And there is a little bit of a difference between how flagellum and cilia function on eukaryotic cells. So we are going to discuss that momentarily. For both of them, they are projections and they're used for motility or some type of motion. On the left, we're seeing a very cute, seemingly fuzzy protozoan that is covered in these little tendrils. This is cilia. So the green bits that are kind of looking like hair, that is the cilia. While on the right, we have a cell that is going to have flagellum. So this is going to be a cell that does have multiple flagellum, but a very classic eukaryotic cell that you can think of with a flagellum is a sperm cell. So let's talk about the cell walls of eukaryotes. There are cell walls on some of our eukaryotic cells, but not all. For example, protozoans and animal cells do not have cell walls. So protozoans have a flexible layer called a pellicle, and sometimes a glycocalyx is also present. If you remember that guy, it's going to be the capsule that we found on prokaryotes. Other structures or other cells in eukaryotes that can have a cell wall include algae, fungi, and yeast. In algae and fungi, this cell wall can contain cellulose, but for fungi, it can also contain chitin. And chitin is going to be kind of similar to what's found in insect exoskeletons. So it's going to be a harder outside, a well-designed structure for support of the cell. And all of these components of the cell wall for eukaryotes are going to be carbohydrates or polysaccharides. So they are not peptidoglycan like what we find in prokaryotes. Remember that the most identifying feature of a eukaryotic cell is going to be the nucleus. So I would star or highlight this slide. It is very important. It is the most important organelle, prominent organelle as well. It's the one that you can see. Other organelles are typically too small to see underneath a microscope, so these guys are super important. It is separated from the cytoplasm by the nuclear envelope. And this envelope is going to be important because it's going to be containing pores so that macromolecules and ribosomes and things can enter the nucleus or exit the nucleus. The nucleus is going to be the site of RNA synthesis. So this is where that central dogma of DNA transcribing into RNA is going to occur, and then the RNA is going to escape through those pores of the nuclear envelope, and then go through the process of translation in the cytoplasm. But outside of the nucleus. It is important to note that the cell itself can be invaded or, have something come through the plasma membrane of eukaryotic cells, but the nucleus is going to be very selective about what can come in the nucleus. So it's going to be very VIP, if you want to think of it that way. So we're really going to be staying away from animal cells in our types of eukaryotes, but we are going to talk about a lot of other types of eukaryotes like fungi, algae, protozoans, and helminths. Helminths is probably as close to animals as we're going to get, and those guys are going to be our little parasitic worms. Let's start with fungi. And the first thing we need to get across here is that fungi are not plants. They do not use photosynthesis. Instead, what they do is break down organic material from decaying organisms. So what this is called is being a saprophyte. So these guys are going to break down decaying material, organic material for their nutrients. And please make sure that you know for all of these examples whether or not they cause disease. That is something I want you to know. These guys do cause disease. They can, for sure. So fungi is going to also include yeast, molds, and mushrooms. So let's talk about at least two of these examples, yeast and molds. We're not going to really talk too much about mushrooms, unfortunately. All right, we are starting with yeast. This is one of our examples of fungi. They are unicellular, so they are a single cell organism. They are oval in shape and widely distributed in nature. We have been able to harness their abilities, and what they provide for us is that we can make bread and alcohol using the process of fermentation, which is super cool. And they can reproduce asexually through binary fission or something called budding. Now, we've talked about binary fission, but what budding is it's like binary fission, but it looks a lot like bubbling. So if you look at the image to the right here, we can see that bud forming off of the parent cell. So it literally looks like a new cell is just bubbling off of the parent cell. Next up is our good friend, the mold. I do want you to know the difference between yeast and mold, and it really comes down to two differences, and that is reproduction and their structure. So when it comes to molds, they are not unicellular, they are multicellular and filamentous. The industrial significance of molds is the production of things like cheese, super duper important, and other things like antibiotics like penicillin. And I do want you to know that these guys can cause disease. They can definitely affect human health. To think about if you have black mold growing in your house. If your house is way too humid or you have water damage, black mold is a very big concern. So yes, definitely molds can cause disease. So mold reproduction is going to come in two different ways, but more often than not, we're going to see mold reproduce asexually. This is going to be a very different process than binary fission or budding. So this is going to be a process where you have spores from a single parent organism that result in a genetically identical organism or offspring to the parent. This is, again, different than binary fission because it's not a unicellular organism. This is a multicellular organism that is reproducing asexually, okay, through spores. Then we have a sexual form of reproduction, where the haploid nucleus of donor cells penetrate the cytoplasm of a recipient cell, and those two nuclei are going to fuse to form a diploid nucleus. Remember that the terms haploid and diploid refer to the amount of DNA within the nucleus. So haploid is going to be half of the set of DNA, and then diploid's going to be the full set. So 2 sets, right? So in this sexual reproduction of mold, it's essentially going to be like a modified version of meiosis. But what it results in is haploid sexual spores that create a diploid nucleus and a genetically unique offspring. So, sexual reproduction. Very different than yeast. All right, moving on to dimorphic fungi. Let's look at that first word and break it down. Di meaning two, and then morphic meaning shape or form. So we're saying that these fungi have 2 forms. Okay, so this is fungi that can grow as a mold or a yeast. This is that 2-form process, all right, so dimorphic. They're kind of interesting. These come in 2 flavors as far as how they can affect humans, so pathogenic or non-pathogenic. The pathogenic is going to be the ones that make us sick or can make us sick, and then the non-pathogenic are going to be the ones that typically do not make us sick. It has everything to do with temperature change. So for the pathogenic dimorphic fungi, it's because our body temperature affects them and causes that change in their morphology. In the non-pathogenic, it's going to change with carbon dioxide concentrations. So these typically are not going to change because of us if we get infected with them. All right, moving away from fungi and onto our next type of eukaryote, and that is algae. These guys are also not plants. Super duper make sure that you know that. These guys are going to be mostly aquatic and also modal, which is kind of fun. These guys are going to be like oxygen scrubbers, if you want to think of them that way. They're going to essentially fix most of the carbon dioxide into oxygen on the planet. So these guys are very important. In fact, the presence of them is what produces most of the oxygen in the atmosphere. Is that not the neatest thing you've ever heard? You thought it was trees. I bet you thought it was trees. The rainforest is important, but algae is where we get most of our oxygen. So this is where 80%, 80% of Earth's photosynthesis occurs is in algae, not plants. So, it's important to save the trees, but it's also important to save the oceans as well. So keep that in mind. Little PSA there. They're also a very important producer for aquatic food chains. So a little bit of ecology here for just a second. They do reproduce asexually through binary fission, but they can also reproduce sexually, so producing unique offspring. So these algae can be unicellular, and there are 4 categories of our algae. The first category is diatoms. The second is dinoflagellates. The third is euglenoids. And then the last is green algae. We can also have multicellular algae, and that's going to come in 3 categories: red, green, and brown. And you might be thinking, but we just talked about green algae. Yes, I know they span the 2 categories. I'm sorry. That is just the way that it is. Just make sure you know that. So we have unicellular algae and we have multicellular algae. So remember that our algae are going to photosynthesize, but again, they are not plants. That means that they are going to convert light energy into food energy. And in order to do this, we need to talk about the wavelength specific pigments that are contained in our different algae. So the first one you've probably heard of. These are chlorophylls. So you have type A, B, and or C that can be included in our algae. These guys are going to typically be green. The main pigment is green. And their big purpose is to convert that light energy into food energy. Okay. Next is carotenoids. These are going to be a few different pigments, such as yellow, red, brown, and green, and or green. So the big important part of this particular type of pigment, wavelength specific pigments, is that it's going to be a protector, kind of like a built-in sunblock. This is very important to our algae. And then we have phycobilins. I'm probably saying that wrong, but I'll be real with you guys. This isn't my flavor of science. So if I say that wrong and you're like, oh, that's definitely wrong, don't judge me too harshly. I know a little bit about a lot of things, but when it comes to wavelength specific pigments, it's not my strongest area of science, so don't judge me too harshly. But these are going to be accessory pigments like red and blue. So this is going to collect light to pass to the chlorophyll. So they're kind of like, not the primary players, they're kind of an assist, if you want to think of it that way. All right, guys, let's talk about lichens very briefly. These guys are some of the most interesting combinations of organisms. It's a partnership. So a lichen itself is not really its own individual organism. It's a combination of organisms that live together mutualistically in a community. Okay, so we have the combination of a fungus and an algae or cyanobacteria. So that is the combination. It's either it's a fungus, definitely have a fungus, and it's either an algae or a cyanobacteria. So when it comes to the fungus, this is the dominant partner. It's going to provide the home or the house, if you want to think of it that way, while the algae or cyanobacteria, whichever one we're talking about when it comes to this particular or these particular lichens, they're going to provide the food, if you want to think of it that way, through photosynthesis. So the fungus is the house, and then the algae or the cyanobacteria are going to provide energy through photosynthesis. So they live together because they are stronger together. They can withstand much harsher conditions if they form this partnership as a lichen rather than living separately as just a singular fungus on its own or an algae or a cyanobacteria. So together they have this partnership that really works for them. So here we're seeing a lichen structure illustration where we can see the fungus forming that house, and then we've got the algae living inside of it, which, that nice little mutualistic community living together in the house and providing the food. On to our next type of eukaryotic cells or organisms. We've got protozoa or protozoans. These are going to be the largest of all microbes. They're pretty big because they are their own organisms. They are unicellular, but each one individually is its own organism, kind of like a individual cat. Mittens is mittens. These individual cells are like its mittens, right? So one cell is its own individual thing. These were coined by Leeuwenhoek, if you remember him from chapter 1, as being the first animals. Because of the way that they behave, they're a little bit odd. They are hunters. So they hunt each other, they hunt small organisms, they can eat algae, which is kind of neat. But yeah, they're mostly aquatic, mostly modal, and we can divide them into even more groups. You probably just heard mittens just then. Either way, so protozoans are going to be our next group, and you do need to know this chart that's coming up next. Okay, guys, so these are the 4 classes of protozoans, and they are based on their motility. So we have flagellates, amoeboids, sporozoa, and ciliates. The type of locomotion, so locomotion is going to be how they move, is going to make sense why they are called what they're called. So flagellates have, shockingly, a flagella. Remember, that's that whip-like tail. And I do want you to know if they can be parasitic, and some of them can. Exciting. Our next class is amoeboids. These guys have something called a pseudopodia or false foot. This is kind of neat, and very few of them are parasitic, but some can be. And if you want to imagine the movement, if you've ever seen the movie Flubber, that's kind of how they move, is like the little green blob in the movie Flubber. Next is sporozoa. These guys do not have locomotion, and all are parasitic. And then we have ciliates. Shockingly, these guys have cilia, those little hair-like extensions, and at least one is parasitic, to humans at least. There are 2 processes that we want to talk about when it comes to protozoans. The first is their reproduction, which is mostly going to be asexually, so through binary fission, budding, or schizogony, which is going to be when the nucleus divides many times before the cell divides into many cells all at once. So that's going to be that image at the bottom there. It's kind of like doing a ton of work like ahead of time and then dividing it up all at the last minute so that you feel really accomplished. But you did the same amount of work. It's just you didn't have to make each individual thing and then move on to the next one, and then move on to the next one. You just made all the nucleus all at the same time, and then distributed those nuclei, which is kind of a smart process. But that's only one of the 3 types of asexual reproduction that our protozoans will go through. And then also some of them do reproduce sexually, similar process to meiosis. 2 parent cells, they produce a unique offspring. So nothing super interesting other than that schizogony. And then we need to talk about encystment. Encystment, when it comes to protozoans, is not dissimilar to bacteria producing endospores. So a cyst is going to be a non-reproductive structure that is highly resistant has a dormant survival form. So again, similar to those endospores when it comes to bacteria, this is going to occur during poor environmental conditions. And once conditions are favorable again, then our protozoan is going to return back to its normal state. So it is a vegetative state until it goes back to an active form. So, encystment. We are in the home stretch of our eukaryotes. So this is the last type of eukaryotic organism that we are going to talk about. And of course, it has to be helminths. These guys are worms. You're probably going to hate these guys. They are super interesting, but also they should make you feel squeamish and kind of have a little bit of an ick kind of feeling. So they include things like tapeworms, flukes, and roundworms. And if you're like, I don't know what a fluke looks like, it's the little pink guy in the middle there. I'll show you another illustration of this guy here in a second. The adults usually are large enough to see with the naked eye, while their babies, larvae, eggs, and some other stages, maybe some intermediate stages, may be harder to see, and you would need a microscope to see them. Not all worms are going to be parasitic. Many are living in soil and water and just doing their thing, living their life. But there are parasitic helminths, and they spend quite a bit of their existence in the gastrointestinal tract of animals, which makes sense. That's how they get around. So let's get a little bit more specific with our helminths. When it comes to this broad term flatworm, we're talking about the phylum Platyhelminthe, which is going to divide into two groups, cestodes and nematodes, and that's where we get the terms tapeworm and fluke. So those guys are like cousins, if you want to think of it that way. And overall, the broad term for those two guys is a flatworm. What classifies them is that they are thin and often have segmented bodies, as you can see in that little illustration where he has all those lines dividing him up. Those segments can pop off and create problems, more worms, things like that. Not a good time. Often those segments contain eggs. So if that segment breaks off, well, you're breaking off like a little package of tapeworm eggs. So gross, love it. From there, we've got our other classification of helminth, and that is roundworms. There's their phylum. You don't have to know it, but it's interesting that it's there. These guys are going to be the little death strings, if you want to think of them that way. They're elongated, cylindrical, unsegmented. They're very classically worm-like, if you want to think of them that way. So round and worm. Roundworms, yes. Let's give you some nightmares. So here are some imagery of our parasitic flatworms. So remember, this is that phylum Platyhelminthes. If you want to think of the fluke and the tapeworm as being kind of like cousins within that phylum, they kind of are. So what you're going to see on both of these fellers is a sucker. Does precisely what you think it does. It's going to hook onto the intestines on the inside of an organism, and they are going to use that sucker to stay in place, as well as probably get some nutrients from their host. The fluke kind of looks like a leaf, but if it was inside you, you'd be thinking that's not a leaf, that's a worm. And then we can see that segment popping off from our tapeworm at the bottom there. Again, those usually contain eggs, so if that pops off, well, yeah, you got eggs all up inside your intestines, broken off from that worm. The other thing that our lovely little tapeworm is going to have is going to be those hooks and a scolex, which is kind of its head, if you want to think of it that way. I will give you a warning here. The next picture is icky, so view it at your will if you want to. And here we go. I find the image of the little guy kind of like peeking up like, hey, what's going on the glove, to be particularly disgusting. And I know we're in a micro class and we got to be like, yeah, there's a lot of gross stuff when it comes to micro, but man, that guy really, really gets me. 'Cause he lives inside of you. He lives inside your intestines, and that can be a really icky, gross time. Meanwhile, the picture on the left, kind of looks super cute, kind of like a little plush toy, kind of velvety. He's adorable. He's kind of cute. Guy on the right, I don't like how aware he seems to be of like, put me back in, coach. Anyway, so that's the end of chapter 4. I hope you enjoyed it. So that was all about eukaryotic cells, and that's all we'll cover on eukaryotic cells. Again, primarily we are talking about prokaryotes in this course, but we did want to cover at least a little bit on eukaryotes. If you have any questions, remember you can always email me. Make good choices. Happy studying.